Hello students, let us discuss about red blood cells. So red blood cells, they are non-nucleated formed elements which are present in the blood. So they doesn't contain nucleus in their cell. So they are also known as erythrocytes, and red color of red blood cell, it is mainly because of the presence of coloring pigment called as hemoglobin. So red blood cells, they are highly specialized, well adapted for their primary function of transporting oxygen from lungs to all of the body tissues. RBCs, they are larger in number compared to other blood cells types. So when you come to normal values of RBCs, the normal value of RBC, it ranges between 4 to 5.5 millions per cubic mm. Adult male RBC count is 5 millions per cubic mm, and adult female RBC count is 4.5 millions per cubic mm. So when you compare between the males and females, males have slightly higher number of RBCs. Morphology of red blood cells, normal shape of RBC is, it is disc shaped and biconcave in shape. And when you look into normal size of the RBC, the diameter is 7.2 micron, and thickness at the periphery, it is thicker with 2.2 micron, and at the center it is thinner with 1.1 micron. The difference in thickness, it is mainly because of biconcave shape, and surface area of RBC is 120 square micron, and volume is 85 to 90 cubic micron. So what is the advantage of this biconcave shape of RBC? So because of biconcave shape, there will be rapid diffusion of oxygen and other substances into the interior of the cell. The cells can enter into the cell because of their biconcave shape, and larger surface area is provided for absorption of substances as well as for removal of different substances. And because of biconcave shape, minimum tension is offered when the volume of the cell alters. RBCs, they squeeze through the capillaries very easily without getting damaged. So these are the main advantages of biconcave shape of RBCs. So normal structure of RBC, so as we have already seen, the RBCs, they doesn't have nucleus, and they have a special type of cytoskeleton which is mainly made up of actin and spectrin, and both these proteins, they are anchored to transmembrane protein by means of another protein that is called as kallikrein. And you have the hereditary condition that mainly results from absence of spectrin, so that is called as spherocytosis. Here the cell is deformed, loses its biconcave shape and becomes globular in nature. So that is the abnormality of the normal size of shape of the RBC. When you look into properties of red blood cells, the very important property is relax formation. So what do you mean by relax formation? So when you remove the RBCs from the circulation, the RBCs, they pile up one above another like pile of coins. So this property of RBC, that is called as relax formation, and specific gravity of RBC is 1.092 to 1.101, and packed cell volume, it is the proportion of blood which is occupied by RBCs and which will be expressed in percentage. So that is called as packed cell volume. And next is suspension stability. So during circulation, the RBCs, they remain suspended uniformly in the blood. This property of RBC, it is called as suspension stability, and the lifespan of RBC is 120 days. So what happens after 120 days of RBC? So that is called as fate of red blood cells. Generally, the RBCs will be present in our body for 120 days, and after that easily it will get ruptured or fragmented. So RBCs, after the 120 days, they will get easily ruptured and fragmented. RBCs, they pass through the capillaries very easily because of their diameters, the diameter of capillaries and RBCs. But when the RBCs, when they pass through the splenic capillaries, the spleens will have the diameter of spleen capillaries will be very small. So, when the RBCs pass through the splenic capillaries, they easily gets ruptured and fragmented. That is why we call the spleen as the graveyard of RBCs. After the RBCs are broken down or if it is fragmented, so then the RBCs will be split into hemoglobin. So, hemoglobin will be released and hemoglobin it will again split into heme and globin. So, heme contains iron and porphyrin. So, iron it will be reused later by the body and porphyrin it will be degraded into bilirubin and which is excreted as bile from the body. And then you have globin component. So, globin upon hydrolysis it forms amino acids and these amino acids it will be again reused later for the synthesis of protein in the body. So, this is the full story of the fate of red blood cells. And next we will move on to the function of red blood cells. The function of red blood cells mainly is the transport function. It mainly helps for the transport of oxygen and carbon dioxide in our body. So, transport of oxygen from lungs to the tissues, hemoglobin it will combine with oxygen and forms oxyhemoglobin. About 97% of oxygen it is transported in the blood in the form of oxyhemoglobin. For the transport of oxygen we require hemoglobin. So, hemoglobin will combine with oxygen and that is how the oxygen will get transported. And next function is transport of carbon dioxide from tissues to the lungs. Here hemoglobin will combine with the carbon dioxide and it forms carb hemoglobin and 30% of carbon dioxide is transported in this form. RBC contain a large amount of carbonic anhydrase. It will help for the transport of carbon dioxide in the form of bicarbonate from the tissues to the lungs and 63% of carbon dioxide is transported in this particular form. So, about the functions of different functions of red blood cells we will continue in the next section. Thank you.